Welcome back. Today we're going to talk about chromosomes and cellular reproduction. So we are going to spend a little bit of time in the course going over cell biology and the cell cycle, and especially paying attention to what you can see actually in the background of this image. So you can see that these are actually chromosomes that are lined up along what's going to be the metaphase plate, and they've got spindle fibers attached to them so that they can be rearranged. So the reason why we're going to spend a little time on the cell cycle and what the chromosomes are doing is because obviously we're going to be talking about different aspects of the cell cycle as we go through the rest of the genetics course. But then also on top of that, it's important to realize that whatever we're talking about on the genetic level is going to manifest itself at the cellular level. So we all need to be on the same page in terms of cellular biology. So let's start with the idea that many genetic disorders are going to result in some sort of altered cell structure or cellular function. As I was saying a moment ago, any alteration to the genetic material or the genome is going to manifest itself usually at the cellular level. And so that's what this says. It says that the genetic disorders are going to result in altered cell structure or cell function. Here, I'll give you an example. So Gaucher's disease is it's a horrible disease, and so it affects lysosomal function. So what's happening is you have these cells in your body called macrophages that normally eat other cells. In fact, that's what the word roots here tell us. So phage is phag is the word root for basically eating, and so like think of your esophagus, right? The food that you eat goes down your esophagus, and there's a phag word root in there as well. And then these eat large things, so they eat other cells. So we're not talking about just something that comes in through endocytosis. We're talking about on a macroscopic scale. So a cell that eats other cells. And so these macrophages eat other cells normally, as they should, but because of a genetic mutation, they swell up because that mutation causes them to lack an enzyme needed to break down the membranes of the cells that they are consuming. So they can't digest the cells that they're eating. And this is a debilitating disease because these macrophages just keep eating, but they don't digest what they eat. So they just get bigger and bigger and bigger and they swell. And when that happens, that causes bloating of the liver, the spleen, and most notably the bone marrow. That's where it just, so if you want an idea of how bad this disease can be, imagine your bone marrow swelling up to the point where you break your own bones from the inside. You can only imagine the excruciating pain that would cause. And that's just one aspect of it. Your liver, your spleen, your other organs are also going to, anywhere you find macrophages, you're going to find swelling and bloating. And that's going to be debilitating. In addition to Gaucher's disease, you could also have cancer, right? We've already talked about cancer, or at least I've mentioned in general. Cancer is essentially a disease of the cell cycle, so it's worth mentioning here because we're going to go over the cell cycle in this lecture. And so cancer is a disease of the cell cycle. Our cells need to cycle through the different steps through which they have to pass in order to, replicate their DNA and then divide into two daughter cells. That's a very controlled process called the cell cycle. And if you might recall that that karyotype I showed you of the biopsy of a tumor cell of a tumor, I should say. And so you could see tumors, for example, from that karyotype, or in general, tumors are made up of runaway cells, cells that, especially a cancerous tumor, it's growing and dividing at a rapid rate, faster than you can actually properly go through the cell cycle, and that's how you end up with those. I may have referred to rainbow chromosomes. That's how you refer to those. I'm sorry. That's how you end up with those chromosomes that have multiple different parts from different chromosomes, which they should not have. So you have all these problems because you're not cycling through the cell cycle in a timely and orderly fashion. So when I said runaway cells, think of a runaway freight train, right? Basically, if you cut the brakes, then you have no way of slowing down, and then you have basically a disaster waiting to happen. And so the cell cycle is very important. The cell cycle keeps a constant chromosome number in the process of cellular division. So that division could be through mitotic cell division. So we talked about that. Mitotic cell division, that's going to be when your somatic cells, your body cells, so pretty much all the cells in your body, in a living organism's body, a multicellular organism, all of the cells except those cells that produce gametes being, in the case of humans, sperm cells or egg cells. So in that instance, then, the cell cycle maintains a constant chromosome number throughout meiosis. But in both cases, mitosis and meiosis, you need the cell cycle to maintain the proper number of chromosomes as the cells divide and as they go from generation to generation via the fusion of those gametes upon conception, and then a diploid zygote developing mitotically after that. So let's start with some basic cell biology here. So hopefully we're all familiar with the idea of prokaryotic and eukaryotic cells from previous courses, but let me just quickly remind you or refresh your memory. I think I already started to talk about this. You'll hear me saying a lot about word roots, because if you know a few word roots, you can figure out a whole lot of stuff in science. And so pro is before, u is true, karyon is essentially, it makes you think of a little kernel. And so this is before there was a little kernel, and this has a true kernel in the center of its cell. That kernel turned out to be the nucleus. And so prokaryotic and eukaryotic cells differ in a number of different genetic characteristics, not just whether they have a nucleus or not, but that is primarily, that's the biggest thing, the biggest difference between them. And you can see that down here, right? So prokaryotic cells, the nucleus is absent. For eukaryotic cells, definitely present. Prokaryotic cells in general basically just have one membrane enclosed compartment and everything else is in there, whereas eukaryotic cells have multiple membrane enclosed compartments, the most notable of which is the nucleus. And so that nucleus is very important. The nucleus is the vault. It's where the genetic material, almost all the genetic material for that cell is housed. Genetic material is so DNA, while it is spectacular for encoding information, it is also a very stable molecule, but you have to remember that our cells are constantly being bombarded by viruses and other organisms, microorganisms that can possibly inject their DNA or RNA into our cells. So the cytoplasm is a rough neighborhood for nucleic acid. It will learn about transcription and RNA processing, and when we send RNAs out into the cytoplasm on purpose, we armor them up with a helmet, which is a 5' methylguanosine cap, and a poly A tail. You might recall that from mRNAs. But my point is that the cytoplasm is a very dangerous neighborhood for nucleic acid, because we have natural defense mechanisms, because viruses are trying to stick their nucleic acids into our cytoplasm so it can get into our nuclei. So you don't want to have your DNA exposed to the cytoplasm, and so therefore you have a nuclear envelope. Your nucleus is where we house the vast majority of our DNA in eukaryotic cells, and then there's and like I said, it's a vault. There is a double membrane system, which is the nuclear envelope that keeps all of that genetic material safe and sound inside the nucleus. So DNA inside the nucleus, I think I may have mentioned this before when we were talking about karyotypes, you characteristically see chromosomes as this characteristic X shape, right? Because that's the best time to take a picture of them. But that's not what they look like most of the time. Most of the time, they are just long, squiggly messes that are just tangled together in the nucleus. And so there's a tremendous amount of DNA in a eukaryotic cell nucleus. So DNA is packaged in eukaryotes with histone proteins. So imagine that you wanted to get, a human cell has about 6 feet of DNA in every single nucleus. So every single nucleus of every single one of our cells has about 6 feet of DNA. And I'm not exaggerating. It's actually, if you were to take the chromosomes and stretch them out and lay them end to end, just naked DNA end to end, you would line up 6 feet worth of material. And that's a lot of material to fit into a submicroscopic space, right? So I guess I shouldn't say submicroscopic, but in a microscopic little nucleus, right? And so you have to wind up that DNA. And we'll talk about supercoiling and all that, but you have to wind up that DNA. And if you take any piece of string and you try and wind it up really tightly and then you let go, it's going to unwind. So you need to have some sort of binding agent. And so that's what histone proteins are. So the naked DNA is packaged in eukaryotes using histone proteins. And what happens is that the DNA wraps around these histone proteins a few times. And so then you have DNA plus histone proteins. That's what makes up the raw material of chromosomes called chromatin. And so chromatin is the material, and then it is wound up into somes. So there's a word root for bodies of chromatin. So that's why they're called chromosomes. They are bodies of chromatin. So chromosomes. And so simpler, accessible circular genomes in prokaryotes, so they're not as long, and so they don't need to be wound up as much. They are basically they form a nucleoid. these are prokaryotic cells here, and so they form a nucleoid somewhere center-ish of the molecule. I'm sorry, of the cell. And then you could also have autonomously replicating free-form little circles, little spaghettios of DNA somewhere in there, and so those would be plasmids. Now, successful cellular reproduction requires three fundamental events. And so the first and foremost, what you need is that the cell's genetic material must be copied. Obviously, it's going to be useless to basically split into two cells if you don't have enough genetic material for each cell, because then they'll both die, right? You can't work off of half of your genetic material. You got to have the whole thing. And so then the copies of the genetic material have to be parsed to the different daughter cells equitably. So now for a bacterium, this is super easy. A single-celled prokaryote is good. It's got one chromosome. So you make you replicate that, and then you put one on one end, put one on the other end, and then split the cell in two, and then you're good. Each daughter cell has a chromosome. But in the case of our cells, our eukaryotic cells, human eukaryotic cells contain 23 pairs of chromosomes. That's 46 chromosomes. And it's not just any 46. It's not like you could just replicate them and then just say, oh yeah, 46 go this way, 46 go that way. No, you have to have a proper set, actually two sets going to each daughter cell, right? So you have to have a copy of, so a pair of chromosome 1s need to go to the left, and a pair of chromosome 1s need to go to the right. And then left and right being, the two daughter cells. And then you got to do it again. Pair of 2s, pair of 2s, pair of 3s, pair of 3s, pair of 4s to the left, pair of 4s to the right, etc. And you just keep going all the way through the numbered chromosomes. And then, of course, you have your sex chromosomes. Pair of sex chromosomes go left, pair of sex chromosomes go right. And so that is a logistical nightmare. And so the cell cycle that we'll be discussing is actually how we get around this logistical nightmare, or how we actually, basically make this all happen properly. The third most important thing that needs to happen, three fundamental events, would be the cell must divide into two daughter cells. So you have the process of replicating the DNA. You have the process of parsing the DNA so that the two cells, you basically have one cell that has all that DNA separated to either end, and then you can start the division process and end up with two daughter cells. Now, prokaryotic cells, as I've said, they simply just replicate their circular genome in a thing called binary fission, and then they divide, and that's it. Each daughter cell gets one copy. It's self-correcting. If both chromosomes go in one direction, well, that's too much gene dosage. Think of like a medication. Your genetic material is like a dosage, and so too much gene dosage, so that's fatal to that cell. And then the other cell didn't get any genetic material, so that's fatal. And so that didn't work. If it doesn't work out properly, then the cells die off, so it's self-correcting. Whereas if it does work out properly, and you have one chromosome goes to one direction, one chromosome goes to the other direction, the cell divides, you have two daughter cells, each with one chromosome, well, then that's a functional replication. So eukaryotic cells perform the three steps, but in a complex, well choreographed fashion. So these three steps in a complex, well choreographed fashion, because it needs to be highly choreographed for all of those chromosomes to be separated properly. So this is a genetics class. We care very much about what the DNA and the genetics and the chromosomes what the DNA and the chromosomes are doing. And so the nucleus is what we'll focus on first. The nucleus is where it all happens, right? A eukaryotic cell nucleus. So you're looking at, you can actually see the double membrane system there. See the two lines, right? So that's the nuclear envelope going all the way around the nucleus. So a eukaryotic cell's nucleus is its largest organelle. It's enclosed in a double membrane, like I said, a double membrane system. That's called the nuclear envelope. Here's a scanning electron micrograph of the basically the face of the nuclear envelope. So we just zoomed in quite a bit more. And you can see all these pores, these nuclear pores that allow things in and out, very small things, but they allow molecules in and out. So it is a vault. It is high security in the nucleus. And then there's that dense nucleolus also that's roughly in the center of the nucleus. And so it's the site of ribosome synthesis. You got to remember that in the nucleus, you have to make a lot of DNA polymerase and a lot of other enzymes that are needed for DNA replication, DNA repair enzymes. So you got to make some proteins in there. And so obviously you need some ribosomes to make those proteins. And so that's what the nucleolus is. Now, what you're looking at, don't forget, now you might be like, well, that's not in the center. But remember, what you're looking at is a three-dimensional spherical nucleus. And you're looking at only one optical slice that was taken through a form of microscopy where you focus up and down through the different optical planes. And so, it looks like flattened out, and this doesn't look very centered, but it all depends on what we're looking at in terms of the angle and which focal plane we're looking at. And so those dark strands, so you can see lots of dark strands, that would be dark strands of chromatin. And chromatin exists throughout the nucleus. You find chromatin everywhere. So this is the nucleus, and you can see that the chromatin is starting to condense. It's all over the place, right? I mean, it's not like there's nothing here. It's just, again, we're looking at different optical slices. And so in this focal plane, you got a bunch of it here, 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 and then the middle. And so but those strands of chromatin are basically DNA wrapped around histone proteins, so condensed DNA. Not fully condensed into the characteristic X shape, we haven't gotten there yet, but certainly condensed down so that you can actually, fit all of that DNA into the nucleus. And so, like I said, chromatin is the clumps or the threads that you see of DNA complexed with histone proteins in the nucleus. So a chromosome, get your chrome words correct, right? So a chromosome is a some, it's a body, just like our body cells are called somatic cells. It's a body of chromatin. So chromosome is a body of chromatin. And the chromatin is going to be made up of DNA wrapped around histone proteins. And then there's actually another chrome word that we haven't even looked at yet. So when the chromatin, the chromosome, condenses all the way down to its most highly condensed state, the name that you would give the shapes of the bodies that exist when you condense down to your most highly condensed state are chromatids. And so those are these little, longitudinal strands of chromatin that's been highly condensed down to the point where one chromosome looks like this little X shape because it's been replicated. And we'll get to that in just a second. Okay, so here is an image of a nucleus of a cell, and then actually the inset is a nucleus of the cell before, and this is the after. And so, like I said before, chromatin condenses to form chromosomes, the chromosomes that you're used to seeing, as they prepare to divide, as the cells prepare to divide. So here's what a nucleus looks like most of the time. So all that is chromatin. You can't really make it out. It's all tangled together, all these different chromosomes. There's 46 chromosomes all tangled up together in there. And they're basically all over the place. And so we have the ability, as I've showed you in the past, to paint chromosomes, paint DNA, all sorts of different colors with different fluorescent dyes. If you use a particular stain, you could stain all the DNA, all the chromatin one color, so that's all that fuchsia, hot pink color. And then you've got, they added a separate dye to light up chromosome 4. And so here's one copy of chromosome 4. Remember, we have 2 copies, so here's the other copy of chromosome 4 over here. And they're like not really any defined shape. It's just, imagine one really long piece of string that's just tangled up with a whole bunch of, imagine a big ball of yarn that's made up of 46 individual strings of various lengths all tangled together, right? That's the nucleus as you're looking at it. But then at a certain point in the cell cycle, the chromosomes condense down. And we'll go over this in just a little while, but for those of you that might vaguely remember this stuff, during prophase of either, mitosis or prophase 1 of meiosis, the chromosomes condense down to their most condensed state. And so that's when they take on those characteristic X shapes. And so while they might not look like characteristic X shapes, you got your flaming hot Cheetos and you've got your white cheddar Cheetos. Right, the white cheddar Cheetos are going to be the chromosome 4s. This is a good example of a characteristic X shape. So it's already been replicated during S phase of interphase. We'll go over that as well. But right now, what you're looking at is, like, imagine this centromere, this little like pinch point right there, and then you've got one arm going that way, and one arm going that way, and then one arm going this way, one arm going that way. And they're floppy arms. They kind of stick together and then just kind of bent, because remember, these are floppy molecules in 3-dimensional space in a nucleus under a microscope. They've been treated with chemicals, so they're all over the place. And so chromosomes in humans exist as homologous pairs. So they're not identical, but they're homologous pairs. This would be, let's say, this is the chromosome 4 that this particular person received from the sperm cell from their father, and this is the chromosome 4 that they received from the egg cell from their mother, right? And so in all the cells in their body, hopefully there are going to be 2 chromosome 4s. In order to be perfectly healthy, you have to have your both copies of those. And so they are homologous. They carry the same genes, right? Can't remember if I mentioned this in the previous lecture, but we are all the same in terms of our race, right? There's genetically only one race, right? The human race. You can take a healthy human male from anywhere on the planet, healthy human female from anywhere on the planet, and they can have a child together. They're genetically compatible. Our race is not genetically based. Obviously, there are different, populations that have higher amounts of certain alleles or whatnot, and so you can make an argument that there certainly are some differences. But generally speaking, let me put it to you this way: we all have the same genes. Human beings have the same genes. It's just different alleles of those genes that make us look and appear different, or have, different biological responses to different things. So that means that we, human beings, are diploid. We carry 2 copies of each one of our chromosomes. 1 copy we received from our mother, 1 copy from our father, and while they carry the same genes, they don't carry the exact same DNA sequence for every single one of those genes, so they have slightly different flavors of those genes, and so that makes them homologous. Sometimes they're going to have the exact same flavor of the gene, and so they're going to be homozygous. We'll see that term a little bit later on. Sometimes they're going to have the same gene, but 2 different flavors, and so then you end up with, basically heterozygous alleles of that gene. So chromosome 4, this is chromosome 4, this is chromosome 4. They have all the same genes, going top to bottom, all the same genes, but they might have slightly different alleles of those genes here and there. And so that's what makes them not identical, but homologous. So we have homologous pairs of our chromosomes. And so because we have pairs, humans are diploid. The abbreviation for diploid, or ploidy in general, ploidy is a lowercase italicized n, diploid is 2n. Our sperm cell and egg cells, our gametes, are just n, right? They're 1n, but we don't say the 1, so they're n because they are haploid, because n plus n makes 2n. That's how we get back up to diploid. Some other organisms can be, like I worked with a species of frog in a previous, job that I worked in a lab, and they were octoploid. So they had 8 copies of every chromosome. So however many copies of the chromosomes you have is just really a product of evolution. We, human beings, are diploid. And in fact, pea plants, the Mendel's, strain of pea plants that he chose to use, those were diploid as well. So that was very fortunate, because that allowed his work to be, roughly applied to what we are, or to us. And there are lots of other organisms out there that are diploid as well. Okay, so most human cells are somatic cells. Somatic, so they're body cells, so they are non-reproductive cells. The reproductive cells are sex cells or germ cells. And so somatic cells carry 23 pairs of chromosomes. So let's talk about this. I get a lot of students that get confused. Our body cells contain all of our chromosomes. So they carry all of them. That means they even include the sex chromosomes. You don't have to just have your sex chromosomes in your sex cells, your germ cells. Your germ cells carry all your chromosomes, just half as many, right? So you have, instead of 23 pairs, you just have 23 in them. But your somatic cells contain, I take that back. The germ cells are diploid. The gametes they produce are going to be haploid. But here, that's kind of a tangent. So let's go back to somatic cells for the moment. Somatic cells carry 23 pairs of chromosomes. That means 46 total. So 2 copies, a homologous pair of chromosome 1, homologous pair of chromosome 2, homologous pair of chromosome 3, all the way up to homologous pair of chromosome 21, homologous pair of chromosome 22, and then either a homologous pair of X chromosomes for an XX female, or a non-homologous pair of sex chromosomes XY for an XY male. So germline cells, as I was starting to talk about a moment ago, germline cells are involved in reproduction. They give rise to haploid eggs and sperm, and so we call those things gametes, the eggs and the sperm. Gametes have only 1 copy of each chromosome. So the germline cells are diploid, but they replicate their DNA once and divide twice. And so you end up with 4 gametes that are, or at least the precursors to gametes, that are going to have 23 chromosomes each, not 23 pairs of chromosomes like a diploid somatic cell. So gametes therefore have 23 unpaired chromosomes, 23 total, as opposed to 23 pairs, 46 total in somatic cells. I may have mentioned this a few times already. A pair of sex chromosomes in humans determine whether an individual is going to be the wild type for chromosomally male, XY, or the wild type for chromosomally female, XX. We'll talk about shortly, actually, pretty soon. We will talk about how the chromosomes can certainly be, not aligned with your gonadal sex or your phenotypic sex, meaning, excuse me, you could certainly be an XY female or an XY female or an XX male. But we'll get to that a little bit later on. The vast majority of individuals on the planet are XY male, XX means female. And so of the 23 pairs of male somatic cells, one pair, as I alluded to, is not completely matched. The X and the Y chromosome, I think I showed you those in that karyotype from the first chapter. X is a very large chromosome. Y is a very small chromosome. They are comically mismatched in size, and therefore they are certainly not homologous. They do not carry the same genes and different alleles. They are not homologous. They do share, I should say this now, they do share one tiny region of homology just so that they can go through the motions. They can pair up and basically be separated and parsed, just like the other chromosomes that are homologous that depend on pairing up in the process of meiosis. But we'll get to that a little bit later on. So aside from the X and the Y sex chromosomes, all other autosomes, the numbered chromosomes, all the other chromosomes, the numbered chromosomes, are autosomes, and they normally exist as homologous pairs. So let me ask you this. You should be very careful, right? Like, so here's a chromosome, and then that's the ending of one and the starting of another. So some of these are lined up kind of end to end. This is not one super long chromosome or anything like that. But is this a somatic cell or a gamete? And so now if you were to count all these up, then you'd probably get a number higher than 23, but it'd be pretty hard because obviously, some of them are bunched up. But here's your telltale giveaway, right? If this was a gamete, then it would be an improper gamete because it has a pair of chromosome 4s in it, right? And gametes are supposed to be just one of each chromosome, no pairs, no homologous pairs. So that's your dead giveaway that this is not supposed to be a gamete. And then if you look at all the other, you count them up, you're going to get somewhere close to the proper number. And what's actually in this image are 46 different chromosomes, or I shouldn't say different, right? 23 homologous pairs, but 46 individual chromosomes. And so this is an example of a somatic cell, not a gamete, because gametes are going to only have one of each chromosome. How about this old-fashioned karyotype that we looked at earlier? So what you can see is that homologous pair, homologous pair, homologous pair, all the way through. So again, somatic cell or gamete, this one's going to be a somatic cell. And so now here's my question to you. Is it chromosomally male or chromosomally female? From what we can tell at the chromosomal level, it's male, right? So this is a karyotype taken from a male somatic cell. Now, I've said this aloud. Here it is in a bullet point and with a little visual aid for you. So the two chromosomes of a homologous pair are usually alike in structure and size. The location of the centromere, we'll talk about what that is in just a moment. So the location of the attachment point, right, the centromere, that's the same. The length of the arms are the same. The position of genes, so gene A is at the same position, at the same what's called locus. However, notice this one has a capital A, which is usually reserved for a dominant allele, and this one has a lowercase a, which is usually reserved for a recessive allele. So we'll talk about dominant and recessive, the dominant and recessive nature of heredity in the transmission genetics lecture after we get through the cell cycle. So the very next lecture. But for the moment, right, what we have is an example of how these are homologous chromosomes. They have the same genes in the same locations, but they might have different alleles of those genes, and that's what makes them not identical. That's what makes them homologous. So one of them was long ago maternally derived, came from the egg cell, so you got it from your mom, and one of them you got from your dad from the sperm cell, paternally derived. And so homologous chromosomes are similar, but not identical, right? Because they can carry different flavors of genes, different alleles. So, I mean, we're really oversimplifying here, but let's say that these two versions of a gene encode a trait such as hair color, right? And so let's say that this is, I don't know, let's go with red hair, right? I guess we go with auburn, or this is auburn hair because it's A, right? And then this would be like blonde hair, right? So it's not auburn, and so that's why it's lowercase a. Whatever the dominant one is usually going to get the letter. Like if this was, like B for black hair, right? Then this would be not black hair. That'd be blonde. That'd be lowercase b. That'd be confusing. So that's a bad example. But basically, you get the idea, right? So the dominant phenotype, the dominant trait usually gets the naming, and then whatever the lowercase letter is just not that. So here we have auburn, and here we have blonde, but that is just lowercase a. Now, I should mention, of course, this is an oversimplification. That was a hypothetical. Hair color is not that simple. All right, so you heard me mention this term called centromere, right? So let's make sure we all are on the same page on what a centromere is. So functional chromosomes have three essential elements. They have the centromere, right? The centromere is that pinch point, right? That's the site of location. We'll get to that in a moment. They have a pair of telomeres, right? Telomeres are quite famous. Many of you may have heard of telomeres in terms of their role in aging. So we'll talk about those in a moment. And then they have to have origins of replication because, of course, you have to replicate your chromosomes. You have to replicate the DNA. You have to take... Well, if you had a chromosome and you condensed it all the way down, it would look like this. However, if you replicated this DNA, yet it still remained attached, then it would look like this if it was condensed down. And so this is a chromatid. This is a pair of sister chromatids. Here's the crazy thing. This is one body of chromatin. This, because they're still attached, is still one body of chromatin. It is what we call, so this is a chromosome, this is what we call a replicated chromosome, but it still counts as one chromosome. And that's very important for when we start to go through the cell cycle for your understanding of the numbers of how many chromosomes, how many chromatids. But for the moment, let's just go over what a centromere is first. So centromeres function as attachment points for the cellular spindle fibers. So you can see here are those spindle fibers coming in, attaching to what are called kinetochore proteins, and they do so at the centromere. Now, the centromere was originally named centromere because it was thought to be in the center because of, I guess, one of the very first chromosomes that were being analyzed. It looked like it was in the center. Turns out it doesn't have to be in the center. And in fact, it's not always in the center. In fact, many times it's not in the center. The human genome does not have a chromosome with the centromere perfectly in the center for any of the chromosomes. But that centromere is important because it is what makes it possible, or the centromeres make it possible for the cell to maneuver the chromosomes. That's how they grab onto the chromosomes and move them around and, like, for example, line them up along what's called a metaphase plate when we get to discussing all the process by which they actually parse their DNA into two daughter cells. We'll talk about the metaphase plate. And so what happens is that the cell attaches these spindle fibers, which are made up of microtubules. So you could just kind of hybridize those two names, spindle microtubules. Sometimes they're called spindle fibers, sometimes they're called microtubules. And so it attaches from either end of the cell, and that's like you grabbing onto this with two hands, one from one arm, one from the other arm. And then you can push with one and pull with the other, and you can move that whatever you're grabbing onto with two hands all around, right? You give yourself a lot of ability to move it all around. And so that's what the cell does. It basically attaches at the centromere, and that gives it the ability to not only move it all around, but also, when the time comes, to pull one of these chromatids in one direction and the other in the other direction. So this whole thing is a chromatid. This is a chromatid, this is a chromatid, and if we were to rip them apart, we would have the equivalent of two different chromosomes, or two homologous chromosomes. Actually, they would be identical, two identical chromosomes, because we made one by replicating the other. And so they're largely identical. Obviously, nothing is 100% perfectly identical, but they're as close as you can get. And so the cell uses the centromere for the point of attachment and also for ripping them apart. So it's used in anchoring centromeres. It's used in anchoring chromosomes to the kinetochore during cell division. The centromere is used in anchoring spindle fibers to the kinetochore, and so that's how we anchor chromosomes and to this kinetochore during cell division. All right, so I mentioned that the centromere is not always in the center of the molecule, or sorry, of the chromosome. And so actually we classify where the centromere is. So it's classified into, centromeres are classified into 4 types on the basis of the location of the centromere. And so you can see metacentric is fairly close to the center of the chromosome. So you have, they look like to be in the middle. They're as close to the middle as you're going to get. So you have metacentric, you could be close to the middle, but a little bit closer to one end than the other. So submetacentric. Then you can be pretty close to one end, right? So you can see this one, that's a clear discrepancy. And so, like for example, like this one, this would be a good example of something that's like submetacentric, right? That's clearly not in the middle. And then you get really, like, look at that one, you get really close to the other end, right? So they're pretty much close, as very close to one end. That's acrocentric. And then when you're basically at the end, right, like at the telomere, then that's why it's called telocentric. So make sure you know metacentric is pretty much in the middle, submetacentric is just off the middle, acrocentric is pretty close to the end, and telocentric is pretty much at the end. The other thing is, whenever you're looking at a chromosome, when you line these things up on a karyotype, like for example, let me say it this way, that chromosome is right side up. And alternatively, let's see if I can find, let's see, I'm looking for, come on, work with me, karyotype. Oh, okay, that chromosome is upside down. How on earth? Oh, that right there. That chromosome's definitely upside down. So that one's right side up, that one's upside down. The only way I know that is because by convention, when we look at these chromosomes, we always put the short arm on top. And so the short arms are the P arms, and the long arms are the Q arms. And an old British group is actually who named the short arm the P arm, and they were trying to be funny in their scientific paper when they published this. And so they said that this was their, as they said, their cheeky joke, which was the short arm is the P arm. P is for petite, and so that's the P arm. And then the long arm is called the Q arm. And do you know why it's called the Q arm? Do you know what Q stands for? That's correct. It stands for nothing. So they said naturally it's the Q arm because Q comes after P, right? And so I guess it was some sort of mind your P's and Q's kind of joke or something like that. But you know what happens when people try to be funny. It's not going to work out. So so basically we just go with it. So the P arm is the petite arm and the Q arm is the long arm. And so the centromere is always shown with, whenever you write these chromosomes, notice like this one's upside down. Notice that you, by convention, you put the centromere in such a place that the short arms are going to be on top. So that's how I know that that chromosome's upside down. So if we were to like cut and paste these images into a karyotype, we would spin that one up, right side up, I guess, in order to show the short arm on top, the P arm, and the long arm is the Q arm. And so then we have, basically at the ends of these chromosomes, we have telomeres. And telomeres are famous because our chromosomes do not do well with age. And so one of the things that can happen is that they, the ends are going to get degraded because they're linear chromosomes. And we have these enzymes that are necessary to help us replenish the ends of our chromosomes, especially during the, it's called the end replication problem. We'll talk about it when we get to DNA synthesis. But our telomeres are very important. We need to maintain the health of our telomeres of our chromosomes in order to make sure that our chromosomes don't degrade more rapidly than they should. And so the telomeres are the ends of these chromosomes. And so they are the natural ends or tips of a linear chromosome. Prokaryotic chromosomes don't have them because they are circular. Eukaryotic chromosomes do have telomeres. And so what they do is they stabilize and protect our ends from degradation. They're sequenced that if you have telomerase enzymes that are functioning well, that's good for your health long term. And in fact, there are laboratory mice that their age has been greatly increased relative to litter mates because of mutations in their telomerase enzymes that made sure that their telomeres remained intact or were actually more robust than they should be. And so that that slowed down the process of aging with them. So understanding telomeres is very important in our study of aging and cancer. And so you might remember I mentioned Craig Venter. And so you don't need to know Craig Venter's story for a test or a quiz. But it's interesting that I mentioned him because he's worth mentioning because he tends to pop up here and there. You might be thinking, like the first time around, I told you how the first time around, people were like, okay, crazy guy, yeah, you're going to sequence the human genome. Go ahead, give it a shot. And then he did. And then the second time, his second act is like, oh, now you're going to synthesize life. Yeah, okay. The first time we didn't believe you, but that was at least feasible. This time, no, you're not going to be able to synthesize life. And then he did. And so, whenever he says what he's going to do, most people laugh at him. But you never know, because he's Craig Venter. And so if I told you that Craig Venter is currently, if you Google him right now, you'll find all sorts of stuff about how he is working on, let me say it nicely. So he's working on aging, but really he's working on immortality, right? And again, you want to say like, okay, this time there's no way. But I don't know. I mean, like he's Craig Venter. He seems to like back it up, and if anybody's going to figure it out, he might be the guy that's going to figure it out. So I don't think he's going to figure it out, honestly. Personally, I mean, we're just not built with parts that can last very long, and we're actually, because I think something I may have mentioned, the fact that we are continuously going to ensure that our genes mutate, our chromosomes mutate over time as an individual, so that for the survival of our species. The downside of that is that those mutations will eventually pile up and eventually lead to cancer. If you live long enough, I don't mean to be a downer or anything, but if you live long enough, you should, if your biological systems are working properly and your DNA replication is working properly, you should develop cancer just from the mutations that you're going to inflict on yourself. So telomeres are quite important and interesting in aging and cancer. I invite you to certainly research more of that on your own if you're interested. We'll come back to telomeres pretty far along in the course when we get to DNA replication and we talk about the end replication problem. Lastly, the third, essential element of a chromosome is the origin of replication. Origins of replication are actually DNA sequences. You can't really see them. They're not easily viewed with microscopy. But if you're going to replicate this chromosome, remember the chromosomes are of different sizes. And so if you have chromosome 1 that's huge and then chromosome, 22 that's tiny, it would be stupid to like just be, you start replicating chromosome 22 and then you're done. And then if you started from one end and had your work away to the other end and chromosome 1, you're waiting on chromosome 1, you're going to be waiting forever. And so we have multiple origins of replication all throughout our chromosomes. So you can imagine one here, one here, one here, one here, one here, one here. Actually, it's quite a bit more than that. And so these multiple origins of replication really speed up the process because, as you'll see in the cell cycle, DNA replication is one of those things that needs to be done properly, and you need all of the chromosomes to be replicated. Like they could start at different times, like you can give chromosome 1 a head start and chromosome 22 could still probably finish before it. But they need to finish at the same time so you can move along in the cell cycle past your next checkpoint. So origins of replication are necessary to make 2 sister chromatids from 1. So the origins of replication are going to make us possible to replicate the DNA in this chromosome and make 2 sister chromatids from 1 chromatid. So now chromosomes in the nucleus are ultimately going to determine what the attributes of the cell are, right? I mean, your DNA is the, it's the material that encodes all of the information necessary to make the tangible assets of the cell. And so that information is going to be contained within DNA, and the stretches of DNA within the chromosomes that are organized into functional units are called genes. And so genes are, as we've said before, the fundamental units of heredity. We had that definition before. But because they are the fundamental units of heredity, they must be passed on. So the information in our genes must be passed on, and this process cannot occur without the cell cycle. We need the cell cycle to pass on our genetic information. And so the cell cycle is basically the progression of the cell through regulated highly regulated processes. And so cell cycle progression is regulated by key transition points called something I just mentioned a moment ago, checkpoints, right? Like, for example, when you're replicating your DNA, you don't want to start it and then just like take a break, right? You got to get that done. If you start it, you got to finish it. And if you finish it, then you move on to the once it's all finished, all the chromosomes are finished, then you've now met the necessary requirements to make that checkpoint, and then you move on to the next phase of the cell cycle. So cells are either growing or dividing. This is something to keep in mind. The number one, so think of cells. Cells have one job, right? You might be thinking, wait a minute, no, there are lots of different specialized cells, lots of different tissue types, and the specialized cells that are differentiated. And so like, cells in your pancreas are secretory cells that secrete all sorts of things like insulin and whatnot. Whereas, like a cardiac muscle cell, it's supposed to just contract to keep your heart beating. But no, actually, what those things are side hustle. The number one job of a cell is to replicate its DNA and split into two daughter cells. That is the number one job, and that's why cells are either growing or dividing. So our cells alternate between two states, basically division and preparing for division. So non-division is when they do all that other stuff, but they're also preparing for cell division. The cell cycle is the sequence of events between divisions in eukaryotic cells. Remember, this is in eukaryotic cells because eukaryotic cells are the ones with all the chromosomes. They're the ones with the logistical nightmare. Prokaryotic cells, super easy, binary fission, replicate, split, done. Whereas eukaryotic cells require the cell cycle, which is a sequence of events, highly choreographed sequence of events that occurs between cell divisions. And so the cell cycle consists of multiple different phases. So you've got, and so we're going to start at about, what is that, like 10 o'clock? So we're going to go clockwise. We're starting at 10 o'clock because why not? And so the longest part of the cell cycle, the largest part of the cell cycle, I can't remember whether I say longest or largest on like a test question, but the most time spent throughout the entire cell cycle is going to be in interphase. And then interphase is broken up into three phases, G1, S, and G2. We'll go over these different phases in more detail in just a second. Let me give you the phases first. So you've got interphase, which is the time between divisions. So here's a brand new baby cell, just got, split off. And so then it has to basically go through interphase to get ready for M phase. M phase, if it's like a skin cell, M phase is going to be mitosis. If it's a germ cell and it's producing gametes, M phase is going to be meiosis. But because they both start with M, we just call it M phase, and then we know that we're dealing with this particular set, which is, so this is what we use to deal with parsing up our chromosomes, or parsing out our chromosomes to indicate, or to leave us with two equivalent daughter cells. And then after M phase, the very last thing is the split, the actual split, and that's called cytokinesis. So cytokinesis is the division of the cytoplasm splitting into two cells. So you've got interphase, M phase, cytokinesis. So let's look at interphase a little bit more in detail. So interphase occurs, like I said, just after the cells have divided, so brand new baby cell. And so if you have a brand new baby cell, well, then you know that it has to grow, just like any baby, right? It has to grow. And so one thing to keep in mind is that the cytokinesis event that just occurred to give us this brand new baby cell, it's not equitable. It's not like you're going to make sure that like 3 mitochondria go, well, it's probably more than that. So like 7 mitochondria go in one direction and 7 mitochondria go to this daughter cell. And they each get equal number of mitochondria and equal amount of Golgi apparatus, equal amount of endoplasmic reticulum, equal amount of ribosomes, et cetera, et cetera. So it's not going to be equitable. So one cell might get like a bunch of mitochondria and not really need to make a lot more mitochondria, whereas this other daughter cell, let's say the daughter cell that we're dealing with right now, didn't get very many mitochondria. So that's one of the things that has to happen during G1. G1 is sort of like building up your infrastructure. Now G1 is called gap 1. So you can see that here, gap 1, but you can kind of think of it as the first growth phase, right? So it's the interval of cell growth, right? Before DNA replication can occur. That occurs during S phase, which is S for synthesis. So in gap phase, gap 1, RNA is going to be transcribed, proteins are going to be made, molecules are going to be made, organelles are going to be built up. You're going to build up your infrastructure. And you're going to stockpile a tremendous amount of high energy nucleotides and other molecules so that you have the energy and then also the raw materials to replicate your 6.4 billion base pairs of DNA that is the human genome. And so That's what happens. You're getting ready for S phase of interphase by going through the very beginning of interphase, which is G1, gap 1 of interphase. All right, so then we do get to S phase of interphase. And so here's where you are replicating your DNA. All those origins of replication, they start to kick on. You've got DNA being replicated this entire time. So DNA is synthesized. And so that's why it's called S phase. DNA is synthesized during S phase of interphase. This is where, so okay, let's put a picture in your mind, right? So this brand new baby cell in the nucleus, you just have a big squiggly mess. And now you get to S phase. And so those big squiggly chromosomes are going to get a sister chromosome attached to them. They're going to replicate them. So you just have this piece of string, and then you just kind of make another piece of string that's still connected to it. So like if you follow the laser pointer, right? So if this is like a piece of string, right? And so here, let's use this. So here's one chromosome, right? And then what's going to happen in S phase of interphase is we're going to make a copying, like a green line right next to it. And so that's going to be our chromosome. So here, let's make a little green line next to it. Let's see if I can do that. So there you go. So there's and the thing is that they're going to be attached. And so now we have I guess we can, that's going to be our let's see, there we go. So that little black thing is going to be our centromere, but that hasn't condensed down. And so to kind of give you an idea, I mean, it's a big squiggly mess, right? So it is not, do not picture this as, I don't have a matching orange, but you can get the idea from this. So here we go. So that's that. And I'm not kidding, like all over the place, right? And so I made that one too long, so obviously you have to have the same there. Let's just pretend those are the same length. Okay, so this is getting away from me. All right, but basically that is a big squiggly mess up there. Kind of looks like a little Subway logo. And so essentially what that means is that, I lost my laser pointer. Yeah, I lost my laser pointer. I am new to this and I cannot get back to a laser pointer. Interesting. Well, all right. Well, I'm sorry to make you sit through that. All right. So, S phase of interphase is where DNA is synthesized, and you make this big squiggly mess. Aha! I have outsmarted this. So, got my laser pointer back. All right, so basically, G2 is coming up next, right? So S phase of interphase, you replicate your DNA. None of it has condensed down yet. And so it's just that big squiggly mess. So if you might recall the image of all those chromosomes where I refer to them as, flaming hot Cheetos and white cheddar Cheetos, the inset, right? The little circle that you saw with all of the chromosomes just mashed together, that is what you should be picturing. So that would be, you've got a whole bunch of chromosomes in there. They're in replicated state because they went through S phase of interphase. And so now G2 is the preparation for cell division. This took a lot of energy and effort and resources. So this is sort of like a little recovery phase, right? So you have another gap phase. And so this is the interval following DNA replication, and the cell prepares to divide. It's got to build up. So M phase, the actual process of dividing, is going to recall, so the condensing of the chromosomes and separating them and then pulling them apart and then dividing into two cells. That requires a tremendous amount of energy. So you got to build up for that. Build up your energy reserves, build up your infrastructure, whatever you utilized. And the other thing to keep in mind is that you do not want to start this process without seeing it all the way through. So that would be G2, preparing for cell division. Now, something that's very important to keep in mind is that not all cells cycle. And so not all cells cycle. Some arrest in G1, so they just kind of sit there in G1. They're not doing anything yet, and eventually they'll go through. And then others permanently arrest in G0. So when I say permanently arrest in G0, when they're in G0, so brand new baby cell, and it doesn't even start anything to make it look like it's in interphase, it just sits there doing nothing, right? So that would be arrested in G0. That means that in G0, it never divides. It's never going to divide while it's in G0. But of course, If it were to come out of G0 and then jump into G1, then it could start growing and dividing and cycling. So a good example of these cells that might be arrested in G0 are your memory cells. And when I say memory cells, I'm not talking about like your childhood memories or anything like that. I'm talking about your immune memory cells, right? Which is obviously very relevant given the SARS-CoV-2 virus. But basically, what your immunity cells are, what your memory cells is that. So the process of immunity is, immunology is very complicated. I'll give you the 5-second version. When you get infected with something foreign, usually it has evolved a way to avoid being detected by your body, but eventually you'll detect it, right? So the goal is to, detect it early on so that your immune system can actually fight it off, as opposed to letting it have like free reign of your body and replicating itself to the point where it can overwhelm your resources and your system. And so eventually it's going to be recognized as foreign, and then you have these cells that are going to eat the foreign virus particle or whatever it may be. They're going to digest it. They're going to display parts of it on the outside of their cells. Then other cells in your immune system are going to look at those and be like, oh, okay, so that's what we're fighting. And they're going to reverse engineer. This is the process of reverse engineering. But they're going to look at that and they're going to make antibodies that fit exactly onto that molecular structure of whatever that portion of the virus particle or whatever it may be that they encounter from those other cells. And so then these cells are now capable of producing antibodies. All of this is taking time. All of this is while whatever it is, you haven't really started fighting it yet until you start producing antibodies. And so the process of reverse engineering the particle to antigen and then making antibodies against the antigen takes time. And so it would be absolutely at this point, you're good, right? So now you have cells that can make antibodies. Let's hope that it's not too late. And so your cells start to ramp up production. You make a whole bunch of these cells that produce antibodies. You make a ton of antibodies. The antibodies opsonize. They basically like coat the foreign particles, and they light them up by saying, hey, this is foreign, this needs to be destroyed. And then other cells see that, take that out, and destroy it. So it would be stupid if the cells that knew what that was and knew how to make antibodies against that particular antigen were to just die off after that. What happens is that some of those cells, when you're replicating these cells to make antibodies, some of those cells arrest in G0, and they're your they're your immune memory cells, right? And so those are the ones that I mean arrest in G0 and never divide. And so obviously over time, if they're just sitting there and when you get exposed to the same thing, then those cells kick on right away and they're like, hey, I got this, I know what that is, and they start producing antibodies and you produce more of those cells and you can have a faster response. That's the whole idea behind a vaccine or an inoculation or immunity given like being exposed to something beforehand. And so that way you can have a much quicker immune response. And so this time around, you don't have to go through that whole reverse engineering process. And so you might not even notice that you were infected again by the same antigen. Or if you are, then it'll be a much quicker recovery. And then the third time around, you have even more of those because you kicked on those cells and you saved some of those cells from both rounds. So that's what the idea behind a vaccine and then a booster shot is for. So you get a vaccine that is usually a subunit vaccine, the vaccine that they're working on currently for the COVID-19 disease, the SARS-CoV-2 virus particle. It's a subunit vaccine. You want to take a little portion of the virus, not the whole thing, not the virulent stuff, just the outside protein coat. You just take a little portion of that. You produce a whole lot of those little outside spike proteins. In the case of SARS-CoV-2, they're called spike proteins. They're these little trimeric proteins, and you produce a whole lot of those, and then you can basically inject those into somebody, and theoretically, then that would be registered as foreign, and eventually you develop antibodies against them, right? And so then when you get infected with the real thing, they're going to have the spike proteins on the outside, the real virus particles. And so then you have antibodies that you can make against those. And so over time, these cells that are arrested in G0 certainly can die off. Like imagine you had like you got an immunization when you were a kid, and now you're like, 60, 70 years old. Well, by then, if you were never exposed to it again, then the cells never had a chance to, kick out, get up into the cell cycle and divide, and so they died off. And so you need a booster shot, right? Or you need to be re-inoculated because you don't have any of those immune memory cells. And that's why you hear a lot of, when there, so SARS-CoV-2 is a different beast, but when there are generally like flu or something more common, you usually hear about like it's very dangerous towards the very young, right? So infants, because they haven't had a chance to have it, build up immunity to anything, and the elderly, because the elderly are usually they certainly don't have as many resources because they're not young and healthy like say like a 20-something-year-old, and then on top of that, whatever immunity they might have very well could have essentially died off, and so they don't have that immunity. And so that's why vaccines are so important to our society, because not everybody's going to be immune. Any vaccine, you're still going to have like it's 80% efficient. So, you're going to get the vaccine and still, there's still a chance you're going to get whatever the disease is. But the more people that have it, there's this thing called herd immunity, where the more people that are vaccinated, the more people that will be able to fight this thing off and eradicate it from their system before becoming, contagious, and so it won't spread as much, right? And so that's the idea behind basically sheltering at home and staying away from one another, and our social distancing rules is right now we have a novel virus that nobody has immunity to, and so because it's brand new on the scene, and so therefore we can't rely on herd immunity or anything like that. We just have to kind of isolate to try and stop the spread of it. So then we finally, so that was all interphase. Interphase is the longest part of the cell cycle. Sometimes I refer to it as the largest part of the cell cycle, which it's both. And so you have G1, you have S phase of interphase where DNA is replicated, you have G2, and then you have M phase. So mitosis prepares cells for division. We're going to start with mitosis. So mitosis prepares cells for division by distributing DNA. So we're talking about somatic cells. Mitosis is for somatic cells. Somatic cells that are going to replicate themselves. They're going to clone themselves. Your skin cells, for example. If you just rub your fingers on the back of your hand, you just rubbed off microscopically a ton of skin cells, and it's no big deal. You'll make more, and you will make more through mitotic cell division. So what we have are replicated chromosomes. They are replicated because they went through S phase of interphase. And so the way we would say this is we don't have, so remember they're still attached to one another, right? So when you take a chromosome and you replicate it, there's a whole other chromosome that you just made a copy of this one. You made a copy of it, but it's still attached. And so therefore it counts as one body of chromatin, one very large chromosome. And so while there's twice as much DNA there, and therefore there's the equivalent of two chromosomes there, still counts as one chromosome. So we have to have a way of saying what we have. And so we say replicated chromosomes, or sometimes you'll see duplicated chromosomes, are going to be denoted 46 times 2. Now you don't say 92. Don't do the math. This isn't a math problem. This is saying 46 that are in their times 2 form, their replicated form. And so you don't say 92 because you don't have 92 individual pieces. You had 46 pieces, and now you have 46 pieces that are twice as big. And so that's why it's 46 times 2. So now these replicated chromosomes must be given to two daughter cells accurately. So you're going to take this 46 times 2 and you're going to turn it into 46 and 46. And so that is what happens, what needs to happen for proper cellular function. So mitosis, or M phase, occurs in 6 arbitrary phases. And I'll give you an easy way to remember these 6 arbitrary phases. Now, if we were in a conventional lecture, you would probably have a better feel for my personality by now, and you might already have somewhat of a feel for it. And that is that I don't take a whole lot of stuff seriously. I really enjoy teaching this class. I love this stuff. I find it fascinating. And I remember vividly things that helped me when I was first learning it, because I was so engaged and so interested in it. And I don't expect everybody to be as psyched about it as I am, but I did find a few things that helped me tremendously to remember things as I was going through my undergraduate and graduate school, or graduate career, I should say. So let me tell you how I learned the 6 arbitrary phases of M phase, and it's an easy way to memorize it. You'll never have any problem. So we'll go over them, and I'll give you the logic behind each step, because I feel like that's easier to remember than to just brute force memorize that, this happens at this point, this happens at that point. But before we even get into the different phases, I had to learn the order of the phases. And I was fortunate enough to have, if you've had any other classes with me, you may have heard this before. I have had had some amazing professors when I was in college. I had a very good college experience. I loved almost all of the professors that I had for all the different classes that I had to take when I was in school, and I went to Boston College for undergraduate. And so one of my professors was hilarious. And so he was, I'm an older individual, so this was a while ago, but he had done a lot of drugs in the 60s, and he certainly liked to tell us about it too. And so, but he was absolutely brilliant too. So one of those, right? Like a brilliant kind of space cadet kind of character. And there were times where we'd be in this huge lecture hall. I had him for a very large lecture format. And so like 300 students, stadium seating, that sort of 9 boards, right? Like 3 banks of boards where he'd write on it's chalkboards, because, this is before we had whiteboards. And so he'd write on the chalkboard, and then he'd like slide it up to the top, and then he'd write on the next one, slide that up to the middle, and then write on the next one, and just leave that there, and then move to the middle bank. And so by the end of class, there'd be 9 boards filled with stuff. And so I was right about the middle of the middle bank, right? So he's working in the middle of the room, and this is like a 2-hour long lecture, and he's like only an hour in or so. And one of the things you should know about him is that you could tell he did a lot of drugs in the 60s, because he would just kind of zone out every now and then, and just kind of like he'd be lecturing, and then all of a sudden he would just stop and just kind of, like take a blank stare at the wall for a second, and you would think, oh, he's just gathering his thoughts, but it would go on just long enough that you're like, is everything okay down there? And then eventually he would just like kick right back on like nothing happened. So it was a little weird, it was quirky, and you got used to it. But this time he was like mid lecture, he was saying, okay, and so then we get to M phase, and so the phases of M phase, and he just dead stop, and he just kind of looks at all of us, and he's standing facing this big crowd of 300 students or so. And he looks at all of us, and he doesn't say a word for like a really long time. So you could imagine if I was in class, I would demonstrate what an awkward silence would be, and I would just look at you guys for like, like 5 or 10 seconds, and the silence, I mean, people would start laughing, and it would be weird right off the bat. But I mean, we're talking like 30 seconds, maybe it was a minute. It was, it got to the point where it was very uncomfortable. People were raising their hand, they were trying to get his attention, and he was just like a blank stare directly at us. So usually he blank stared at like, the wall or something, and so it was just kind of weird and a quirk. But this time it was creepy. And so he's looking at us, and nothing's happening, and I'm just like, what is going on? And so then he turns around, and I'm like, oh, okay, he's back, welcome back. And so he turns around, he walks to the board, and he starts writing, and so I'm like, oh, okay, we're back now. And so I like start writing down what he's writing, and then I quickly realized that I should stop, and so I stopped. And then like at this point, everybody's just kind of laughing, and people are just trying to figure out what the hell's going on, because he is drawing, and he is a phenomenal artist, but he is with chalk, with white chalk, he is drawing a dog on the board. And it's actually a really spectacularly good drawing of a schnauzer. So you can imagine all the little hair and everything. He was doing a spectacular job. But this went on for an extremely long time, way longer than this story, that if you haven't fast forwarded me yet. And so basically he, he's drawing this dog, this schnauzer, and I mean, we're talking, he's putting he is like street artist going into this thing. And so he's putting like, I don't know, I can't remember. I feel like it was like, 5 to 10 minutes of him just drawing. And at this point, I mean, people are trying to get his attention. They're raising their hand. They're calling for him. They're saying, hey, professor, excuse me, professor. And he's like, oh, nothing. Other people, I mean, after like a minute or two of this, people started getting up and leaving. And he's still committed. He's into it. And I'm sitting there. I had another class in the same building, like basically right after this class. So I'm going nowhere. And so I'm just like, well, we're going to see where this is going. And so I was just, kind of watching what he was doing, and I'm watching the dog, and I was actually marveling at the level of detail. It was pretty impressive. I vividly remember what it looked like right now. And for me, this was, well, I won't say how many years ago this was, but it was many years ago. And so basically, and he spent a long time on this. And I mean, at some point, finally, we're talking minutes and minutes into this. He finally gets to the end of the dog. Like he started at the head, right? And he's going left to right on the board, and he gets to the end. And I mean, it's a good sized drawing too. It's like taking up a good chunk of the board. And of course, he finally gets to the dog, the end of the dog. And he puts the last leg up, And then he grabs a yellow chalk and he draws this little like arc, and then it like basically this little puddle. And so the dog's peeing. But what was weird was that not only was the dog peeing, but it looked like he emphasized what the peeing was, because he drew this sort of like honestly, it was a parallelogram. He drew a parallelogram around the like puddle of pee. And so he finally turns around and in a really loud booming voice, he was like, "All right, everybody, all right," and kind of startles some people that weren't paying attention anymore. And he's trying to get the people their attention back, who's left. And there's like probably, if we started with 300, we were probably down to like maybe 200. I mean, like a good third of the class had got up and left. He didn't care. And so he's like, "All right, calm down." And everybody's talking and laughing and just having a great time at this point, because it's just like the craziest thing. And so, and half the class isn't even paying attention to him. So he just spends all this time to get everybody's attention. And so what he does is he basically says, "All right, okay." So, and he points to the board and he says, "Well, what is that?" Right? Like smartass number one is like, "A dog peeing." And he's like, No, what is that?" And so he points to like the shape that's around the puddle of pee. And so like smartass number two is like, "A parallelogram." And then like I remember there was somebody else that was like down in front that said, parallelogram. And then somebody else like started arguing with them. It was like, "No, it's a rhombus." And so they were arguing back. He's like, No. And so then he goes into this like long talk about how basically it's this like, so he said, how like, so I went to Boston College, and so he goes into this rambling, tirade about how when it snows a lot and the snow plows come and they push the snow up onto the sidewalk, if you live up in a high rise, if you want to walk your dog, and he said that was his dog, and I don't remember the name of his dog, and he's like, when you want to walk your little dog, then like it's hard because there's all this snow everywhere and it's just too hard to walk your dog, and so you could go to the pet store and you can buy these, and so he's pointing to that shape around the puddle of pee. And he's basically like, "You can buy these. These are absorbent mats. These are absorbent mats that absorb the urine." And at this point, I'm seriously wondering, like, if the poor man is having some sort of medical episode or something like that, if I should call 911. But he's like ranting about how you can buy these absorbent mats and how wonderful they are and how much they cost. And he says, you can buy these mats that are really absorbent. These pee-pee mats. Prophase, prometaphase, metaphase, anaphase, telophase. Pee-pee mats, right? And so I'm sorry for taking so long to tell you that story, but I tell you, from that point on, I had it memorized. I knew that exactly the order was prophase, prometaphase, metaphase, anaphase, telophase, because it's pee-pee mat. Of course it's pee-pee mat. And I know this works because when I'm walking around campus, I get random students yelling from like, way too far away. They're like, "Hey, Dr. Babu, pee-pee mat." And so apparently it is, something that's easy to remember. There is no need to yell it at me. That a simple wave is good enough. So anyway, so there are 6 arbitrary phases to mitosis. There are prophase. Some textbooks don't show prometaphase. I'll talk about that. I'll show you what I'm talking about with that. But they kind of bleed into each other. You can see here it just shows prophase, metaphase, anaphase, telophase. But prophase, prometaphase, metaphase, anaphase, telophase, and then the 6, they're arbitrary phases, right? So once you're done with M phase, some people include cytokinesis and other people don't. So there you go. So cytokinesis would be when you get the actual splitting of the 2 cells, because like I said, they all just kind of bleed into one another. It's not like there's a clearly defined point at which this phase ends and this phase begins, except for actually 1 or 2 of them. So one of them definitely has a clearly defined point when it has ended. But other than that, they're all just kind of like, they just kind of bleed into each other. So what we're going to do is go through the different phases of mitosis first. So we'll start with prophase, then we'll get into what prometaphase is, then metaphase, anaphase, telophase, and then of course cytokinesis. I'll show you a little animation to try and drive it all home. We'll go through it first with static images and I'll describe what's happening. And then I'll show you the animation and that'll kind of drive it all home. And what's nice is that then you can contrast that with the M phase, that is meiosis, which occurs in germ cells to produce gametes, the nuclei for gametes. In gametes, you don't want 46 and 46 in 2 daughter cells. You want 23, 23, 23, 23 in 4 daughter cells.